Just a minute, some network issues there, just 2 minutes. I request the AB team.
असतो मा सद्गमय तमसो मा ज्योतिर्गमय मृत्युर्मामृतं गमय ॐ शान्ति शान्ति शान्तिः। या कुन्देन्दुतुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा।
Now I request Honorable Vice Chancellor sir for the floral welcome of the Chief Guest of the occasion. Thank you, sir. Now, I request Dr. K.N. Guriprasad, Director, SVIS, and Dr. Kavita Sharma, Professor and Head, Department of Chemistry, for the virtual welcome of the resource person. Dr. K. Venkatechalam, Professor, Department of Analytical Chemistry, University of Madras, Chennai. Dr. Mohammad Aslam, Professor, Department of Chemistry, NIT Srinagar. Dr. Shankar Narayan, Professor Emeritus, Department of Advanced Material, AC College of Technology, University of Madras, Chennai. Now I request Dr. Kavita Sharma ma'am, Convener, FDP, Professor and Head, Department of Chemistry, SVIS, SVV Indore, to throw some light on Faculty Development Program.
Thank you, Divya. Honorable Vice Chancellor of Shri Vaishnav Vidyapeeth Vishwavidyalaya, Indore, Dr. Upinder Dhar sir. Chief Guest, Dr. Bal Subramanian, Professor Emeritus, University of Madras. Eminent speakers, Dr. Shankar Narayan, Professor Emeritus, Department of Advanced Materials, AC College of Technology, University of Madras. Dr. Mohammad Aslam, Professor, Department of Chemistry, National Institute of Technology, Srinagar, Jammu and Kashmir. Dr. K. Venkatachalam, Professor, Department of Analytical Chemistry, University of Madras. Dr. Santosh Dhar, Rector and Dean FDSR, Shri Vaishnav Vidyapeeth Vishwavidyalaya, Indore. Dr. Niranjan from Metron Private Limited. Dr. K. Guruprasad, Director, Shri Vaishnav Institute of Science, Shri Vaishnav Vidyapeeth Vishwavidyalaya. Coordinator FDP, Dr. S. Venkatesan, Associate Professor, Department of Chemistry, SVIS. All HOIs, HODs of all the constituent institutes of Shri Vaishnav Vidyapeeth Vishwavidyalaya and dear participants. On behalf of Department of Chemistry, Shri Vaishnav Institute of Science, Shri Vaishnav Vidyapeeth Vishwavidyalaya, Indore, I welcome you all in this first faculty development program on recent advances in analytical chemistry. New technologies have helped us a lot in this COVID-19 phase and still to continue teaching and learning activities so that all academicians, industrialists, researchers are able to interact with each other across the globe virtually. I would like to share that Department of Chemistry, Shri Vaishnav Vidyapeeth Vishwavidyalaya is organizing its first faculty development program. Thanks to technology that Honorable Vice Chancellor Sir is with us from US and Dhar Madam is also with us. Thank you Sir and Ma'am for giving your valuable time, guidance and support. Faculty development program plays a significant role to update themselves in diverse skills and abilities. Faculty development program help faculty members to deal with the rapid changes and shifting paradigms in subject areas, education patterns, and innovations in teaching and learning technologies. I would like to thank Dr. Bal Subramaniam Sir, Chief Guest of today's program, and to all eminent speakers for accepting our invitation to share their knowledge and experiences with us. In recent years, the automation of analytical instruments such as autosamplers, databases, data treatment, and control of instruments using microprocessors has accelerated the advancement of analytical chemistry techniques. Automation has long been employed to solve a number of industrial and laboratory problems. Much of our daily life is dependent on chemical analysis. Accurate quality control analysis ensures the quality of food we eat, the medicine we use, the water we drink, and the air we breathe. Among the sciences, analytical chemistry stands out as a practically versatile, useful, and important field. Today, we would like to start this FDP by remembering one of the legends, Isaac Moritz Payet Poltov, who spent 64 years as a faculty member in the In this faculty development program, we have 6 different sessions in which today, along with this inaugural session, we will be learning about nanomedicine from indigenous natural product, enhanced delivery of curcumin by nanoparticles in breast cancer cells by respected Bal Subramanian Sir. On the second day, that is on 19th April, we will get to know about applied concepts of analytical chemistry and gas chromatography, principles and application by Dr. K. Venkatachalam. Sir will be continuing on 20th April on ultraviolet spectroscopy and sample analysis by classical methods. On Thursday, 21st April, we will be knowing about recent developments in spectroscopy techniques by Dr. Shankar Narayan Sir. Sir will also be discussing the recent developments in spectroscopy techniques. On 22nd April, we have scheduled special session for online instrument demonstration by Metron India Private Limited. Thank you, Dr. Bal Subramanian Sir and Dr. Niranjan Sir for arranging this session for us. On 23rd April, Dr. Mohammad Aslam Sir will be speaking on thermoanalytical techniques a powerful characterization tool for material scientists and chemists, which will be followed by a valedictory session. So before I close, I would like to share that I have recently joined Department of Chemistry as professor and head. Although I was associated with SVITS since 1996, we are university since 2015, and I was working with SVIFS, Shri Vaishnav Vidyapeeth Vishwavidyalaya, Indore from 2015. And recently, Honorable Vice Chancellor Sir has given me this opportunity to work with this chemistry department. Also, as head Center for Women Studies. Thank you, sir. I would like to share that Chemistry Department, Shri Vaishnav Institute of Science, Shri Vaishnav Vidyapeeth Vishwavidyalaya offers BSc Chemistry Honors, MSc Chemistry, MSc Analytical Chemistry, and MSc Environmental Sciences. So without taking much time, I welcome you all once again, and I'm sure this FDP will be highly beneficial to all of you. Thank you very much. Happy learning. Over to you, Divya.
Thank you, ma'am. Thank you so much. Now I request Dr. K. N. Gulprasad, Director, SPIS, SVV, deliver the welcome address.
Thank you, Dr. Divya. Chief guest for the inauguration of Faculty Development Program on recent advances in analytical chemistry, Dr. Balasubramanian, Professor Emeritus from University of Madras, Vice Chancellor, Sri Vaishnav Vidyapeeth Vishwavidyalaya, Dr. Upinder Dhar, Rector and Dean FDSR, Dr. Santosh Dhar, Dr. Kavita Sharma, Head Department of Chemistry, faculty colleagues, participants, and guests. I, at the outset, welcome all the guest speakers who will be joining us in this faculty development program starting from today on various aspects of analytical chemistry. In science, chemistry becomes the fundamental science for every other branch of science. I work in the area of biological sciences, and we understand biology as a complex chemical reactions regulated by natural forces. And what we try to understand is how these chemical reactions are naturally regulated, and that makes it biology. Even if it is physics, the behavior of the matter will be fundamentally dependent on the nature of the matter. Whether the substance will have the properties like magnetic properties, or will it have an electrical conductivity properties, or will it have a resistance to work as an insulator? All these properties will be determined by the nature of the substance. So the first thing in science becomes chemistry, since we need to analyze the chemical nature of anything which we are going to study, because the behavior of that will depend on the chemical nature and the chemical reactions which are possible under natural conditions. Analytical chemistry, as Madam Kavita explained, will be exploring the new techniques of particularly the computer techniques and microprocessor techniques in making the instruments more efficient in analyzing the chemical reactions and also chemical nature of the substances. So any advancement in these analytical tools will not only advance the knowledge of chemistry, but it will also help in the advancement of the knowledge of biochemistry and biology, and also the material physics in the process. So all those who are joining this faculty development program will be benefited by these talks and also demonstrations of certain instruments, irrespective of the area in which they will be working, since science has become an interdisciplinary area for a long time now, and chemistry is a fundamental area that is required for all branches of science. With this introduction, I welcome all the guests and resource persons and the participants who will be participating in this faculty development program. Thank you. Over to you, Dr. Divya.
Now I request Honorable Vice Chancellor sir to address the gathering.
Dr. S. Subramanian from University of Madras, Dr. Venkatachalam, all other distinguished resource persons for this program, Dr. K.N. Guru Prasad, Dr. Kavita Sharma, all other colleagues from the university, and all the participants of this program. I would like to congratulate the Institute of Science and specifically Department of Chemistry for organizing this faculty development program. And I would say that it is very timely because keeping in view the National Education Policy 2020, challenging times are going to come. As we know that NEP 2020 is a very flexible policy, which is a very student-centric policy. But at the same time, in the next 15 years, it will change the face of higher education in the country. There are many changes which are going to take place. Say, one of the important change that is going to happen, and in fact, it has already started, that is a multidisciplinary approach. That means during the times to come, we cannot afford to restrict ourselves to a particular discipline. Say the big challenge that will be for the teachers will be that they shall have to teach not only those who have foundation in say in the case of chemistry, those students who are having foundation in chemistry, not only to teach those students, but teachers will have to teach even those students who do not have background in chemistry. Because under this multidisciplinary approach, students will have the freedom to choose their courses. Maybe there may be a student in physiological psychology or comparative psychology, and he or she may opt for a course in chemistry. Therefore, challenge is to design the curricula in such a way or design the courses in such a way that even those who do not have a foundation in chemistry can also understand certain courses. So there are going to be two types of courses, one for those who are from the same domain and another set of courses for those who are not from the same domain. So therefore, teachers have to be up to date. Further, faculty shall have to be ready for teaching short-term courses and longer duration courses, both simultaneously. This is so far what we have been doing. We prescribe a course, a 3-credit course, 4-credit course. So you have to teach during the semester and that's it. But now it is going to change. We may have to offer certain even short-term courses, maybe 2-credit courses. Therefore, there is going to be redesign of curricula, redesign of the courses. So that's again, it becomes challenging for the teachers, but we have to do it because it is for the betterment of the students, and also for enhancing the quality of education. Times are going to come when students will be studying certain courses through virtual platforms, say MOOCs, and they will come to the class for clarification of the concepts. If a teacher is not up to date, he or she cannot clarify the concepts. So again, this again a challenge. That's why to cope with such challenges, faculty development programs are very, very essential. Earlier, we used to have once in a while, okay, you may attend that program in a year or maybe 2 programs in a year if you wish. But now there is no scope for wishing to do that. One has to attend the faculty development programs continuously. Higher education institutions will have to organize faculty development programs continuously, and faculty shall have to come forward and remain up to date in the discipline chosen by them. Therefore, we need to prepare ourselves for such challenges. And so far as the theme of today's faculty development, which we are starting today, faculty development program is concerned, it's analytical chemistry. See, as Professor Guru Prasad said, it is a foundation. It is one of the foundation disciplines of science. And whichever the discipline one chooses, student chooses, he has to have the scientific temper. That's why I said that there may be a student from a totally diverse discipline, but he or she may choose a course from chemistry. See, analytical chemistry has been important since the early days of chemistry, providing methods for determining which elements and chemicals are present in the object in question. During this period, significant contributions to analytical chemistry included the development of systematic elemental analysis by Justus von Liebig and systematized organic analysis based on the specific reactions of functional groups. The first instrumental analysis was flame emissive spectrometry developed by Robert Bunsen and Gustav Kirchhoff, who discovered rubidium and cesium in 1860. Most of the major developments in analytical chemistry took place after 1900 AD. During this period, instrumental analysis became progressively dominant in the field. In particular, many of the basic spectroscopic and spectrometric techniques were discovered in the early 20th century and further refined by the end of the 20th century. The separation sciences follow a similar timeline of development. In the 1970s, many of the techniques began to be used together as hybrid techniques to achieve a complete characterization of samples. Starting in, say, around 1970s, into the present day analytical chemistry, there has been, we have seen progressively becoming more, this analytical chemistry progressively becoming more inclusive of biological questions, say bioanalytical chemistry. Whereas it had previously been largely focused on inorganic or small organic molecules. Lasers have been increasingly used in chemistry as probes, and even to initiate and influence a wide variety of reactions. the late 20th century also saw an expansion of the application of analytical chemistry from somewhat academic chemical questions to forensic, environmental, industrial, and medical questions, such as in histology. Therefore, that makes this analytical chemistry as a very important and upcoming field. In recent past, in recent decades, a lot of work has been reported in the scientific journals. So therefore, a faculty development program in this area is very timely, and our faculty need to be up to date in this field, which has further scope for growth. So I wish all the best to all the participants attending this program, and I'm sure that they will feel better off by the end of this faculty development program. And once again, I welcome all the resource persons to this faculty development program organized by our university. Thank you very much.
Thank you so much. I'm sure this will go a long way in understanding the concepts of analytical chemistry. So now I feel privileged to introduce to this gathering our chief guest on the occasion, Dr. S. Balasubramanian, Professor Emeritus, University of Madras. Dr. S. Balasubramanian is Professor Emeritus and Head, Department of Inorganic Chemistry, University of Madras, and former Director, Center for Advanced Material Research, Well University, Chennai. Sir has pursued his PhD from University of Madras in 1985. Sir is having 43 years of research experience and 30 years of PG and UG teaching experience in Madras and Anna University. The area of interest of Sir that are microcyclic chemistry, macromolecular chemistry, environmental chemistry, weather chemistry, nanochemistry, and photochemistry. Sir is having the more than 100 international publications and national publications, and on his name, he's having 9 papers. Sir is also the Secretary of Royal Society of Chemistry and a faculty advisor of American Chemical Society. Sir is a member of American Association of Advancement of Sciences and an affiliated member of IUPAC. Sir is a fellow of Indian Chemical Society and Institutions of Chemists and a life member of Society of Advancement of Electrochemical Science and Technology. Sir is a life member of Indian Society for Radiation and Photochemical Sciences and a life member of Indian Society for Analytical Scientists and Indian Science Congress Association and Chemical Research Society of India. Sir is also the president of Spectrum Chemical Society of School of Chemical Sciences, University of Madras. The project funded under Sir, that is a total amount more than 10 million under the Department of Science and Technology, Department of Biotechnology, Defense Research and Development Organization, University Plan Commission, and Industry. Sir has awarded many prizes, like the award awarded as the Commonwealth Academic Fellowship in 2010 at the University of Oxford, awarded RSC Service Award in 2015, elected as Fellow of Royal Society of Chemistry, Editor-in-Chief for Journal of Neuroscience Letters, elected as a Secretary of RSC South India and Faculty Advisor of American Chemical Society. Such published articles in the Delta Transaction as one of the top 10 articles in 2008 and in Inorganic Chemical Communications, which is as a top 25 article in 2005. There is reviewer of many, more than 20 international journals and a chartered chemist and representative of the Royal Society of Chemistry. Sir is also elected to the board of Research Director, American Biographical Institute, and citation in prime journals like Journal of American Chemical Society, Annual Review Chemistry, Coordination Chemistry, etc. Sir is having the MLU in Stanford University in the area of environmental sciences, biographical sketch and Marquis WHO in the Science and Technology, American Biographical Institute, National Maritime Loan Scholarship, and travel grant from RSC to attend the International Material Chemistry Conferences in University of UP. Sir is invited speakers in the Indian Science Congress Associations and chairman of one of the sessions of the India's chemistry organized by DRDO in Agra. A member of board of PG studies in the chemistry in the Chennai and participated in e-learning workshops in the UK and first investigator to get the industrial consultancy project from the School of Chemical Sciences. Such is the resource person of the academic staff, College, University of Madras, and team member of CLRI in the implementation of ISO 9000 for Kizar Hussain Tannery for one of the most modern tanneries in India. Hosted RSC team from Cambridge, UK, to deliver the lectures on RSC publishing, and Sir is having the CSIR coordination chemistry section in Chennai Science Festivals. Sir has visited many countries like USA, UK, Germany, Japan, Italy, France, Switzerland, Egypt, Singapore, Malaysia, Hong Kong, Dubai, Thailand, Sri Lanka, etc. Recently, Sir has visited California, Los Angeles as in the Advanced Functional Material Conferences as a chairperson and in the ACS National Meeting and invited to the National University of Singapore, then plenary speaker in Biopolymer Conference last week of Vegas, and sixth symposium Stem Cell Cancer Research event in Taiwan. May I now request you, sir, to address the gathering on the topic nanomedicine from indigenous natural products, enhanced delivery of curriculum by nanoparticles in breast cancer cells. Over to you, sir.
Thank you for. Am I audible? Yeah.
Yes, sir, you're audible.
Yeah. Good morning to all of you. First of all, I would like to thank the respected Vice Chancellor, Dr. Upinder Dar, and the Dean Santosh Dar, and the HOD, Dr. Kavita Sharma, and Dr. Prasad, and Dr. Venkatesan, who invited me for this conference. And as a Secretary of the Royal Society of Chemistry South India section, I would like to take this opportunity to thank Sri Vaishnava Vidyapith Vishwavidyalaya, one of the oldest universities in Madhya Pradesh, for this faculty development program. And I'm really honored to be the chief guest of this event. Is it clear? My voice is clear? Yeah. So it is nice that Dr. Dar, Upinder Dar, even though he's in US, maybe in his late midnight, so he's kind enough to be here amongst you to give the presidential address. And it's really interesting that the chemistry department has ventured into this faculty development program. So one of the mandates of Royal Society of Chemistry is, in addition to the publication and the honors to the members and hosting conferences, seminars, and in addition to that, the one is CPD, continuous professional development. This is a very important concept in every field of science, technology, and engineering. Like many of you who have visited the Western countries, you may be aware of the fact that even the doctors, if they don't get the CPD, if they don't get the credit, even with a very high qualification, they cannot continue their practice. So it is a must. That means people working in each and every field of science, technology, medicine, and engineering and other branches should update themselves so that they can deliver the goods without any issue to the stakeholders. So that is the most important thing. As a part of the continuous professional development, the UGC has earlier organizing the refresher course and the orientation course for all the faculty members who are inducted in the university. So unlike school education where they have formal degree in education like B.Ed or M.Ed, the faculty who join the university, they don't have a formal training in education or particularly in the education technology. So the moment they join, they have to undergo a 3-week orientation course, which should be followed by a refresher course. For each and every stage of their career promotion, the attendance and the participation in the refresher course is mandatory. The same thing is continued now as the faculty development program, which is a mandatory for the faculty in all disciplines, irrespective of whether they are in science or engineering or medicine or whatever the case may be. So in fact, there'll be a break after this, isn't it, for the inaugural session, for my technical session? Yeah. So I will cut short this because I'll be giving the first talk as a resource person. So the interesting the theme what has been taken for this event is the recent advances in analytical chemistry. So basically, I'm trained as an analytical and inorganic chemist in the University of Madras. Then I branched out to inorganic chemistry and did all my research in synthetic inorganic and subsequently in nanomedicine and nanotechnology. So that is our research area. So the moment I joined analytical chemistry, my professor gave the definition of analytical chemistry in a nutshell. If you can answer two questions, then that becomes analytical chemistry. What are those two questions? What and how much? These are the two fundamental questions. If you can answer, then you become an expert analytical scientist or analytical chemist. So you should know what is the sample given to you. What is that you are analyzing? That is point number one. That's called qualitative analysis. You simply say, oh, this is an organic compound, this is an inorganic compound, this is polymer, this. So you just identification of the molecule or the compound given to you. This is the answer for the first question. What is it given to you? Next one is, it is not simply enough to say what is that you have. You should also be able to tell how much you have. When you have a mixture of compounds, you should be able to tell how much each one of the component is present in your mixture. So you should have a methodology or a technique to analyze whether it is in a larger quantity or in a micro quantity or in a nano quantity. So you should have the technique. Am I audible? Yeah. So you should have the technique. The problem becomes more and more severe when you go down the scale, when you are going from milligram to microgram, microgram to nanogram, nanogram to picogram, picogram to femtogram. So you need to have extremely sophisticated analytical technique and also the reagents which are all of very high purity. So then only you will be able to quantify not only in parts per million. People usually talk about ppm, parts per million. Not only in parts per million, you can also talk in terms of ppb, parts per billion, and parts per trillion, ppt. So in fact, earlier one of the topics I used to give to the faculty development program is ICP MS. So inductively coupled to plasma spectrometry coupled with the mass spectrometry, where you will be able to analyze the compound and express its in concentration in parts per trillion. So why the question arises, why do you want to analyze a compound or a material which is in such a small quantity? You know, the countries like US, USSR, and many other countries send space, including our own ISRO, sends spacecraft to the other planets. They collect the samples from there, and you have to analyze. Then the sample is brought back to the Earth. Then we have to analyze what are the components present. Some of them may be in milligram, some of them in microgram, and some of them may be in PPT. So unless and otherwise you have the technique to analyze, you cannot identify such a compound is present in your sample. That is why it is very important to master the instrumentation technique and also update your knowledge to analyze the various options available, both in destructive as well as non-destructive analysis. Because if the sample is very less, You can't do the analysis in one method and exhaust the sample and say, sorry, I don't have any more sample to carry out. No. If you know a non-destructive means of analyzing the sample and quantifying it, yes, you can do it. Okay. So both destructive and non-destructive analysis are very important in analytical chemistry. And also people talk about lab on a chip. So the chemistry means you see a wet lab and an instrument lab and a dry lab where you have a lot of instruments and a lot of conventional methods for analyzing substance by qualitative and quantitative methods. But lab on a chip where you can have a very minute sample and you will be able to analyze. And particularly this is useful in areas like forensic science, where you get a very tiny fraction of the sample where you should be able to analyze and tell what exactly is present. And particularly in crime detection, you don't get a large quantity of sample. The sample quantity is very, very less, and you should be able to say what is it and how much is it. So in this context, this is very important that all, and as Dr. Guru Prasad said, analytical chemistry is not only branch, it pervades everything. Like omnipotent, God is present everywhere. So same way, the analytical sciences, analytical chemistry is present everywhere. Whether you do chemistry or physics or biology or material science or engineering or medicine, you should be able to tell what and how much. So this is very essential for all the scientists and engineers and doctors practicing all over the globe. So this is where the role of analytical chemist becomes very essential in all branches of science. And I am really glad that the Vaishnava Vishwavidyalaya, which is one of the oldest universities in Madhya Pradesh, is organizing this event. I wish all this program a success. And I once again take the opportunity to given to the Royal Society of Chemistry as a secretary of the Royal Society of Chemistry South India section to take part in this event. And I thank you all for listening to this program. Thank you. Come on now.
Divya has some problem in connectivity. What is the issue?
Venkateshan sir. Hello, Venkateshan sir is invited for words of thanks.
Yes. Thank you, sir. Thank you so much for your kind words of wisdom. It's a great pleasure for all of us to listen to you. Thank you once again. Now the session is carried out with a question answer. If anyone is having any questions, you can ask. Raise hand.
Venkateshan sir is invited for words of thanks.
On behalf of Chemistry Department, Sri Vaishnav Vishwavidyalaya, I would like to propose the vote of thanks. First of all, I would like to propose hearty vote of thanks to eminent chief guest, Professor Dr. S. Balasubramaniam sir, Professor Emeritus, Secretary, Royal Society of Chemistry, University of Madras, who in spite of his busy schedule, accepted our invitation and has came here to this online event, grace the faculty development program. Thank you, sir, for your interesting and thought-provoking lecture. And I'm sure that all the participants have taken note of your valuable research ideas, key points, and suggestions. I place on record our deep sense of gratitude to Honorable Vice Chancellor, Professor Upendra Dhar sir, for delivering the presidential address and being a backbone in motivating all research activities, conducting conferences, workshops, and webinars in Sri Vaishnav Vidyapith Vishwavidyalaya. My heartfelt thanks to Dean Dr. Dhar Madam, Sri Vaishnav Vidyapith Vishwavidyalaya. I extend grateful thanks to Director, Sri Vaishnav Institute of Science, Dr. K.N. Guruprasad Sir, for the valuable insights and sharing your perspectives and motivation. I also extend my sincere thanks to Dr. Kavita Sharma Madam, Head Department of Chemistry, Sri Vaishnav Institute of Science, for the keen interest, guidance, and active involvement to make this faculty development program from paper to reality. I also extend wholehearted gratitude to other resource persons, professors, Metrom India Private Limited executives, Association of Chemistry Teachers, and Chemistry Department Organizing Committee, technical team, technicians, and faculty members of Sri Vaishnav Vidyapith Vishwavidyalaya, and faculties of other universities, colleges, and dear students. Thank you, one and all.
Thank you, sir. Thank you so much. May I now request the gathering to raise for the national anthem?
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you. With this, we come to the end of the session. Thank you all.
We will be going to start session 1 of Dr. S. Balasubramanian sir with the same link after 10 minutes of break.
Okay, best of luck.
Thank you, sir. Thank you, sir, for giving us your valuable time. Take rest, sir. Thank you. So all the participants remain present. With the same link, we will be starting our session within 10 minutes. Thank you. Can we start, sir?
Sure.
Yeah. Hello, Divya.
I have it's muted.
Hello,
Just
Divya.
unmute. Just unmute.
Unmute.
May I now request Dr. S. Balasubramanian sir to address the gathering? Can we start, sir?
Sure. So, good morning once again to all of you for this first session. So, you might have noticed the topic is, am I audible? Yeah. So, the topic is slightly different from the theme of the event. But I chose to give in this because we have been working in this for quite some time. And the as I said earlier, I used to give talk on the ICP-MS, which is a very nice technique where one should be able to analyze the material in parts per trillion. So that's what I used to give. But that is, of course, it is more of a instrumental technique which is relevant to this. But since there are other participants who will be talking about this, and instantly we have arranged one lecture on ion chromatograph also, where you will be able to analyze the ions in the sample, and it will be a live demonstration on request. Metro India was magnanimous enough to give this demonstration. So this is basically, as I said, ours is a synthetic chemistry group, and we have been working in the area of synthetic inorganic chemistry, synthesizing organic compounds as well as inorganic compounds. And subsequently, we moved to the synthesis of polymers. Then we decided to move on to the biocompatible polymers, that is biodegradable and biocompatible polymers, which can be used as a carrier for drug delivery. So if you have seen the topic of my lecture, this is it's a nanomedicine delivery by of indigenous material. What is this indigenous material? We have chosen curcumin. This curcumin is nothing but the active principle from turmeric, which is used in our day-to-day cooking. In fact, each and every one of you might have come across turmeric in your daily food, and also as a cosmetic. So people in particularly countries like India or China, they are particularly in Southeast Asian region, they are quite aware of the turmeric and its efficacy in our life. But many of you may not know, it is not only a food material, it can also be used as a therapeutic agent if you are able to extract the active principle from turmeric and use it for treatment. So this is what we are going to see now.
Sir, your slide is not changing.
That's what I'm trying to, this slideshow, I'm just trying to get the first one. Somewhat it is frozen. Just wondering why. It's not. Should go to slideshow mode. No problem in getting the slideshow mode. So it's not. Shall I proceed this way? Or,
Yes,
from
sir.
begin-
Please.
yes, Professor. Or in the from beginning, you can start, Professor. In the file, in the left side, left hand corner.
Yeah, file is there. Yeah.
From
File,
the beginning, the file.
no, that's what, from the beginning, that's not showing. That's what I'm saying. When I go to slideshow.
Okay, Professor.
Please continue, sir, with the
Continue,
same.
Professor. Continue, Professor.
Wait. Okay, fine. Maybe I'll manually do that. I'm not sure why it is frozen. Okay. So you can see the slide now. Slide is visible.
Yes, sir. slide
Yes,
is
sir.
visible.
Yes, Professor. Yes.
Yeah, fine.
Yes, sir.
So no, as I go to slideshow, from the beginning it should come. That is somehow it's not coming. I don't know why. That's a problem.
We are able to see it, sir. Please.
Yeah,
continue.
fine. So I'll go ahead with this and see the curcumin, which is the active component of the turmeric, can be for various ailments. And earlier it was considered as an antiseptic, but later people have found that it can be used as an anti-cancer agent also. If you are talking about a drug, it can be given in various forms. The slide is visible properly. I think this.
Yes, sir.
Yes, sir.
Professor.
It's
Yes,
visible.
Professor. Yes, Professor.
So if you say if you are taking a drug, it can be a pill or it can be a capsule or it can be a syrup or it can be an injection. So there are various ways of taking a drug. The conventional method of delivering a drug to a living system has some problems. Like there are some inadequacies, like less bioavailability. You have to have a higher dosage, and there is a problem of systemic toxicities, and also inadequate drug concentration at the site of infection, and you need to give a treatment for extended period of time. And whereas the ways in which you can administer drug also varies. It can be intravenous, oral, buccal, sublingual, rectal, intramuscular, transdermal, subcutaneous, inhalational, topical, ocular, and there are other routes also. So by depending on the route of administration, the efficacy of the drug also changes. For example, if you are taking a pill and the same medicine is given by an intravenous injection, the efficacy varies, even though the constituent is same. So the routes of administration also plays a very vital role in the therapeutic efficacy of a drug. And so that is where, but when you are taking a drug, you don't have a control over it. You just take it. You take a pill or a capsule or any liquid medicine. So it straight away goes into the system and gets absorbed. You cannot really control the way in which it is getting into the system. But that is where the controlled drug delivery came into the picture. Because usually, if you go to a doctor and you have a viral fever or a bacterial fever, so the doctor will administer a drug. Okay, you take this medicine twice a day and maybe three days or a 5 days course. So the each dosage, you may be taking 50 milligram or 100 milligram of the drug, right? So how this is determined? Of course, this is the minimum quantity required to eliminate all the microbes which causes the problem in your system. Suppose if you are taking thrice a day and 5 days a week, so you don't leave any trace of the microbe which has attacked your body, and you completely eliminate them. That is called the therapeutic dose. The therapeutic dose, what you actually take, is more than what is actually required for eliminating or killing the microbes. The reason is the drug is not only attack the microbe in a particular site of attack, it goes to the entire body, as a result of which you need to take more than what is actually required. Whereas in the case of controlled drug delivery, you can release the drug as and when it is required, and also in a targeted manner also. So the advantages of controlled drug delivery is controlled dosage, increased bioavailability. I'll come to this term bioavailability in a subsequent slide. Biodegradable carrier, it has a high stability, high carrier capacity. That means it incorporates many drug molecules in the particle matrix, and you can incorporate both hydrophobic and hydrophilic drugs, and you can administer through various routes of administration, like what I mentioned in the earlier slide. It is target specific, and it has a reduction in side effects. This is one of the most important things. When you take medicine more than what is actually required, you not only get cured from the illness what you have, you also get into the problem of side effects. Okay, so that will be taken care of if you take the exact quantity what is required, and that is achieved by controlled delivery. Okay, so how can you deliver the drug in a controlled manner? You encapsulate in a polymer. You always think polymer is toxic to the human body, but it is not true always. But there are polymers which are biodegradable and biocompatible. So these polymers are used as a vehicles or carriers to carry the drug and release it in your body in a controlled manner. So these are all some of the examples, like polylactide, polyglycolide, polylactide, copolymer glycolide, polyanhydride, polyarthoester, and polyepsilon caprolactones. These are all some of the biodegradable polymers. Of all this, the most popular and the FDA approved one is the third one, PLGA. So this is the one where we started working on that, and we could people normally buy it from a commercial vendor. It is available commercially, one can buy it, but we didn't buy it because we are basically synthetic chemists, so we could synthesize it in our laboratory with the various composition, with various molecular weight, with various ratio, and with the different hydrophobicity and hydrophilicity. So we can have a control over what we are synthesizing, because that is the advantage of being a synthetic chemist. So you can do whatever you want. But if you buy from a commercial vendor, so you have to modify your research according to the product what is available in the market. So that's the difference. And of course, these polymers undergo degradation in your system and gets excreted. It does not stay in your system after the delivery of the drug molecule. That is why they are biocompatible and also they are biodegradable. So no harm is done to the system when you are taking the medicine along with the delivery vehicle, that is the polymer matrix. So what are the factors which affects the biodegradation? One is the chemical structure, chemical composition, and here it's a copolymer. You have the lactate in which you can have the ratio from 1090 to 90. So you can have different ratios. The distribution of monomers, and again the molecular weight. You can have low molecular weight, medium molecular weight, high molecular weight, and again molecular weight distribution, because you know, unlike simple organic molecule, polymer doesn't have a single molecular weight. You might have studied in your master's degree that the polymers have both the weight average and number average molecular weight. This is always a average molecular weight what you are talking about. And also the morphology. It can be an amorphous, it can be semicrystalline, it can have microstructure, and it can also have a residual stresses. And presence of ionic groups. We always think polymers are neutral, but you can also introduce ionic. We have done that, and we have introduced ionic groups in our polymers for not for this, for a different study, and which can be, this is called polyelectrolyte, which will be used for treatment of industrial effluent. Some of my research group members worked on that, and also we use this ionic polymers as ionic liquid for the synthesis of organic compounds. Again, we could get some good publications in the synthesis of organic compounds using ionic liquids. And similarly, physical factors like size, shape changes, solvent-induced cracking, change in diffusion coefficient, and also the mechanism of hydrolysis, absorbed and unabsorbed compounds. So all these factors affect the biodegradation of polymer. And if you look at the PLG, as I said, this is a FDA approved polymer, and this already it is in use clinically, and people are, it's not in clinical trial, it is people are using it clinically, and some of the drugs are already encapsulated in PLGA matrix and given to the patients. In future, we will have more and more drug molecule being incorporated in a biodegradable polymer matrix, so that you will be able to reduce the dosage and reduce the amount of drug you are taking, so that the patient compliance will be there, and also you can reduce the side effect, that is the toxic effects of the compound can be reduced or sometimes even eliminated. The PLGA is a approved polymer, it's biodegradable, biocompatible, and it has increased bioavailability. Of course, when there is a water solubility, the bioavailability also increases and can form nanoparticle. And we have taken different type of drugs for our study. So when we first submitted our proposal to Department of Biotechnology, so the neglected disease in developing countries is one of the neglected diseases, tuberculosis. You don't see tuberculosis in Western countries. Tuberculosis is mostly in tropical countries like India and other Asian countries. And the drug which is given for the treatment of tuberculosis is rifamycin or rifampin, isoniazid, and pyrazinamide. And this all the three, and added to that, the fourth one is also given for the tuberculosis. And leprosy also you give rifampin or rifamycin, both are one and the same. And clofazimine and dapsone, that is a very popular leprosy. So these are all encapsulated in a PLGA matrix, and we have tried both tuberculosis and leprosy drug. And in addition to that, we ventured into two more areas. One is the cancer. Cancer, you have a lot of, you may be knowing, there are three ways of treating cancer. One is chemotherapy, the other one is, second one is radiation, the third one is surgery. So some people get cured by chemo, some people by radiation, some people by the surgery, some people need all the three. Some cases with all the three, the metastasis occurs, this patient may not survive. And you have chemotherapy means all synthetically made drugs like doxorubicin, or otherwise known as DOX, and paclitaxel. They have a lot of side effects. You might have seen people who have undergone chemotherapy for during cancer treatment, they might have had a lot of side effect in their body. But whereas if you are using a naturally occurring compound, then you can avoid the side effect. Some of them are curcumin. This line is from the flowering plant in our country, and the curcumin again obtained from the turmeric, which is used in our everyday cooking. Okay, so we have made use of curcumin for our controlled drug delivery. So curcumin was encapsulated in a nanoparticle, biodegradable, biocompatible polymer matrix, and it is used for the treatment of breast cancer. In fact, breast cancer is the major killer as far as, and a lot of medicines are given, but still the death rate is quite high. So that was the reason why we thought why we should not try out a naturally occurring compound, the curcumin, which is available in plenty in our country. So bioavailability, if you take 100 milligram of, suppose if you say a Dolo or Dolo 600 or any other, 600 means 600 milligram. So if you are taking 600 milligram, you have two things. One is active principle, that is the active component, and the other fillers and other material present in your tablet or capsule. So the active component, let us say it is 500 mg. If you are consuming 500 mg, it does not mean all the 500 mg will be there in your bloodstream. Only some quantity will be there in your bloodstream, and that is what is called bioavailability. This is the fraction of the administered dose that reaches the systemic circulation. Suppose if you are giving an IV intravenous injection, then it is 100%. But if you are taking a oral dose, depending on the extent of the absorption by your system, the bioavailability varies, and this is obtained from the graph time versus the concentration of the drug in plasma. The area under the curve is the bioavailability. For an ideal drug, the bioavailability should be 100%. Meaning, if you are taking 100 milligram, all the 100 milligram has gone to the system. But it doesn't happen always. Most of the time, the bioavailability will be usually less than what you have actually taken. So that varies from drug to drug and from case to case. So we will look at how the PLG, in fact, when we started the work, we could see some literature reference for the synthesis of PLGA. But once people understood that it has a lot of clinical application, and people started only patenting the discovery, and no information was available as far as the synthesis of PLGA is concerned, because it is commercially a successful, clinically proven biodegradable polymer. So people didn't want to simply give the procedure or the protocol for everyone to synthesize. So so these are all patented. So we tried out in our laboratory, and we could synthesize the PLGA from simple monomers, lactate and glycolide, that is DL lactic acid and glycolic acid. And you need to have a catalyst. Usually one can use stannous compound like stannous octoate or stannous chloride. We are used to both of them. and you will have a stepwise synthesis, high temperature and stepwise reduction of vacuum, and you end up with the PLGA, which is the foam-like polymer, which can be stored under refrigeration for over a period of time. For several months, nothing happens to the stability of the copolymer. And this slide, of course, sorry, since I couldn't go to the slideshow mode. So now it has come. I'm sorry. So finally, we started working. Sorry for the technical issue, the earlier thing. So I am showing the slides which I already gone through. Yeah. So this was the last button slide. So now we will see the synthesis of PLGA, and you can see the, thank God that you can see the slideshow. So on the left corner of the slide, you will see the foam-like white foam-like substance that is a solid PLGA, which you could get from the right side where you see the liquid. After the final treatment, you end up with this nice polymer. So this is the synthetic procedure. You have the D-lactic acid, glycolic acid, and here we are taking the stannous chloride as the thing. So as I said, 3 stages. So first you start at 120 degree at a vacuum of 100 mm of mercury, where you remove the water molecule and continue stirring. The stage 2, where they raise the temperature to 150 degree and maintain it for 7 hours at a vacuum of 100 mm. And then you end up with the oligomer, and then you raise the temperature to 180 degree and continue the reaction for 7.5 hours at a very low vacuum of 15 mm of mercury. Then you stir it, cool it, and dissolve in chloroform and add diethyl ether as a non-solvent. You end up with this, and if you keep it in a vacuum desiccator, you end up with a nice foam-like PLGA thing. So this is about PLGA, what we have synthesized. Curcumin, you all know, which is a plant which is indigenous to our country, and people from time immemorial have been using this turmeric. The botanical name is Curcuma longa, and the turmeric is a product. It is obtained both from the root as well as from the stem, and the turmeric used in your cooking as well as the your cosmetics. And if you can extract the active component, actually the turmeric is yellow in color, but if you extract curcumin, it will be orange yellow. It will be more of orange in color. It's a polyphenolic compound, and of course, as I said, curcumin can be obtained both from the stem as well as from the root, and it is more popular in the root. It has a anti-cancer potential. It can downregulate transcription factors like NF-kappa beta, kappa B, AP1, EGR1, and also it can downregulate the expression of COX2, LOX, NOx, NMP9, and UPA, TNF, chemokines, sulfur surface adhesion, and cyclin D1, and also growth factor receptors. And if look at the biological and pharmacological activity, it is antioxidant, anti-inflammatory, anti-cancer, antimicrobial, anti-parasitic, anti-cholesterol, anti-mutagenic. It has a wound healing, cardiovascular, and hepatic and neuronal. It is like one drug for treating various diseases. The one on the right side is a rhizome. In fact, in the next slide, you will see the dry rhizome, which you buy in the market, and the structure is given on the right side. So where the diketo compound is a dione, and it is a dried material which will use for extracting various organic solvents have been used. Of course, you cannot extract with the aqueous media. So organic solvents are used to extract curcumin. It's a chemopreventive. In animal model, it has been proved as a to suppress the carcinogen-induced tumor genesis. and also it inhibits the growth of implanted human tumors in the animal model. And people have carried out clinical trials of curcumin in patients with colon cancer and familial adenomatous polyposis, pancreatic cancer, and multiple myeloma. And all these successful clinical trials have been carried out. And as far as the limitation is concerned, as I already told you, the solubility of turmeric is very less. That is why the absorption in your system is also less. And if you increase the solubility by extracting curcumin from the turmeric, you can increase the solubility and also increase the bioavailability. Still, it is less. You can further increase it by converting it to nanoparticle. So the solubility goes up still further, and the bioavailability also goes up. And the maximum solubility and bioavailability is obtained when you incorporate in a nanoparticle biodegradable polymer matrix. We could achieve a bioavailability close to 95%, which you cannot do it in a simple curcumin or even in nano curcumin. So the limitations with the normal curcumin is the poor bioavailability, it is hydrophobic, and they are interrelated, non-soluble in water. And it has been established by human clinical trials. One can take 12 grams of curcumin per day without any side effect. That's allowed. That is the finding people have reported when carrying out clinical trial with curcumin. So if you just look at the therapeutic potential of curcumin, it is chemotherapeutic. You can use it for cardiovascular disease, multiple sclerosis, diabetes, gallstone formation, chemopreventive, anti-angiogenesis, anti-inflammation, Alzheimer's disease, fibrosis, HIV replication, wound healing, cardiotoxicity, and also for treating nephrotoxicity and antioxidant and cataract formation. Curcumin has been tried out in all this. Of course, the success rate is not uniform in all cases. It varies from case to case, and even in as an anti-cancer agent, of course, depending on the type of cancer, the and also the progression of the cancer, the efficacy of the curcumin also varies. So if you just look at how it works in a patient affected with the cancer. What is cancer? It is an uncontrolled growth of your normal cell. The normal cell, when it is transcripted, it becomes uncontrolled growth. You get tumor cell, it proliferates, you get the tumor growth, and then it gets invaded and get tumor. So the one in the pink color are tumor cells, the normal cells are in blue. So it gradually increases when it becomes tumor, the 100% it becomes a tumor cell. Whereas when you treat with curcumin, the normal cell after transcription, there is only a small number of the cancer cell, and even after invasion, the number is less, and even after the formation of tumor growth, it is relatively less. So these are all the ways in which you can demonstrate how curcumin acts in the human system. Okay, polymer scaffolds. So what are these things? It is nothing but the polymer matrix in which you will be able to incorporate a drug, and also you can release a drug at known rate or a prolonged duration. The drug release occurs basically in two ways. One is the diffusion through pores in polymer structure, and diffusion of drug through degradation of the polymer is the final step. We just look at the mechanism by means of which the drug is released. One is by diffusion, polymer degradation, and combination. The polymer degradation, again, it has three different ways in which it occurs. One is the enzymatic degradation, the other one is hydrolysis, the third one is a combination. The hydrolysis, it can be bulk or surface erosion. This is how the drug is getting released. So, of course, when the surface erosion occurs, it becomes a sudden burst release. But in the earlier, it will be a controlled release. What we need is a controlled release and not uh, uh, yeah, uncontrolled release. That's the burst release. So in our study, what we have done is, many of you know, who are people who have worked in the area of nanoscience and nanotechnology. Preparing a nanoparticle is easy, but maintaining it in the same size and shape becomes rather difficult because they tend to agglomerate or associate and becomes a larger particle. Both synthetic and naturally occurring molecule can be used as a capping agent or a nanoparticle and retain the size and shape of the particle.
Audible. Sir, you're not audible.
They're not audible, sir. Hello.
Yeah. There was a problem with the net and it was reconnected. So that was the problem. So sorry for that. So can you see the slide now and am I audible? Yeah,
Yes, sir.
fine. Yeah, good. So I said there are 4 different type of capping agent. I mean, for naturally, biocompatible capping agents like chitosan, dextran, polyethylene glycol, and TPGS, tocopherol polyethylene glycol succinate. We used it in 2 ways. One is as a capping agent, and another one is an emulsifier. And in fact, when we use TPGS as an emulsifier, we were able to get much higher efficiency in the delivery of the uptake and delivery of curcumin in the polymer matrix. So you will as a PLGA curcumin as a water in organic solvent, you add a curcumin acetone 5 ml. So you use a sonication not as a bath sonicator, as a probe sonicator. So you sonicate it
Not audible, sir. You're not audible, sir. Participants, please wait. This is network issue from sir.
Participants are requested to wait for a minute. There is a network issue. That is joining within a minute.
Venkateshan sir, please call sir. Please call Balasubramanian sir.
Yes, ma'am. I will call, madam. I will call.
All the participants are requested to have some patience. There is some network issue. That is joining soon.
Sorry, there is a interruption, and
Which
now Yeah. So there was a disruption in the net. So after sonicating and lyophilizing, so you will be able to get the dextran capped curcumin loaded PLGA monoparticles. So this is what you see in the slide. The same methodology is followed for the synthesis of chitosan and PEG capped curcumin loaded PLGA nanoparticles. So this slide gives you the method by means of which you prepare the nanoparticle with the various capping agent, TPGS, that is tocopherol polyethylene glycol succinate, chitosan, dextran, and polyethylene glycol. And all this, again, you use TPGS as an emulsifier. So that is also used in the study. So what you see in this slide, where you start with the TPGS and acetone, and where PLGA is 60-40 ratio. In fact, we have done it, synthesized the PLGA with a number of ratios. And in this study, we have taken 60-40, and TPGS taken is a 10:1 ratio. Then you add curcumin, and you sonicate it for 1 minute with the 30 kilohertz ultrasonicator. Then you add dextran and water. So you get again sonicate for 1 minute, and again further sonication of 3 minutes. You after removing the solvent by continuous the stirring and followed by centrifugation at 10,000 RPM for a period of 30 minutes. Then you lyophilize it. You end up with TPGS emulsified dextran capped curcumin loaded PLGA nanoparticle. The same methodology is followed for TPGS emulsified chitosan capped as well as PEG capped nanoparticle. So the methodology is same, only the conditions are slightly different. And if you just look at the mechanism of surface modified curcumin loaded PLGA NPs, you have different capping agents. You have the PLGA here, and the curcumin is there, and the emulsifier TPG is there. And the experiment is done in vitro using a synthetic medium, that is PPS buffer, the phosphate buffer saline, which has a pH of 7.4, which is similar to the physiological pH. So you have the surface modified curcumin loaded PLGA nanoparticle, and subsequently the curcumin is released from the polymer matrix, and the curcumin is released at different time intervals, and finally the polymer erosion occurs, and the control release of curcumin from polymer matrix occur, and ultimately the polymer degradation occurs, and it results in a burst release of or sudden release of curcumin. So this is the methodology. And if you just look at the profile, drug release profile, and if you just plot the number of days the curcumin released versus the cumulative percentage of curcumin drug release, so we could follow it to a period of maximum of 30 days. But when only it is with the capping agent, dextran, curcumin, and PEG and TPGS, so we could see the controlled release up to 21 days. And whereas if it is emulsified with the TPGS, you can see the control release goes up to 30 days. So earlier it was just 20 to 21 days. Now we could see the release up to 23 to 30 days. And you see the raw curcumin where it gets separated in an aqueous medium, whereas the nano curcumin, it forms on the right side. You get a almost a it's like a true solution or a homogeneous solution, where it is completely distributed like any other and so these two graphs show the in vitro cumulative release profile, that is time in days, and versus the cumulative percentage of curcumin drug release. And as I told you earlier, just the capping agents alone, you could see on the left hand side, the release occurs up to a period of 21 days. The same thing when it is with the capping agent like dextran, chitosan, and PEG. You have the emulsifier also with the TPGS. You are able to extend the release up to 30 days. So this really tells you the role of TPGS, that is the emulsifier, in modifying the characteristics of the PLGA nanoparticles. And this table gives an idea how PLGA, that is polylactide glycolide polymer nanoparticle with the capping agent, C for chitosan, P for polyethylene glycol, and D for dextran, and how it varies. So drug loading efficiency varies from 75 to 80, 69 to 89, and 54 to 71. The drug content also varies from 4.6 to 5.5 mg, 4.9 to 6.1, 4 to 5.1. And drug release, as I told you, 20 to 23 days, 21 to 30 days, and 21 to 30 days. And also there is a decrease in the antioxidant activity. So ultimately what you can see is you are able to increase the drug loading efficiency, increase the drug content, and increase the number of days in which the drug is released. And also we have measured the water contact angle. So the below 90° with the water contact angle is there, the material is supposed to be hydrophilic. Above 90 it is hydrophobic, and above 180 it is super 150 it is 180 it is super hydrophobic, and below 30 it is super hydrophilic. So we were able to see a reduction in the water contact angle, meaning that after emulsification, the water contact angle gets reduced from 55 to 31, 79 to 50, and also 75 to 31, meaning the nanoparticle with the drug becomes more hydrophilic. That means it can increase the solubility in the physiological medium and also increase its bioavailability. And of course, this table again gives an idea how in the first three, 79, 75, 55, the theta values become 60, 50, and 31 respectively when you are emulsifying with the TPGS. And also particle size also gets reduced from 180 to 120. This gives you an idea the particle size distribution. As you know, it cannot be a single particle size. There will be a mixture of particles with the different sizes. So left side you have the only with the capping agent, and the right side with the emulsifier, TPGS emulsified nanoparticles. And again, this also the left side you have the with the chitosan cap. So you have the chitosan cap with the tocopherol emulsified thing. So you can see a reduction in its particle size. The see the next table gives you an idea how the particle size, polydispersity index, and zeta potential and water contact angle all varies by introducing a emulsifier. So it becomes there is a reduction in the particle size from 198 to 188, 186 to 165, 194 to 157. Similarly, the polydispersity index increase varies from 0.18 to 0.29, 0.04 to 0.15, 0.01 to 0.08. The zeta potential also increases from 23 to 40, 28 to 40, 0.07 to 34. You know, the zeta potential more positive or more negative, that is plus 30 or plus minus 30. That means it has a much better efficacy when compared to the lower or higher values. And similarly, it is becoming more hydrophilic by reducing the water contact angle from 55 to 31, 79 to 50, and 71.5 to 31 degree. So this gives a TEM image of the TPGS emulsified curcumin loaded PLGA nanoparticle. And it's a SEM image of the surface modified curcumin loaded PLGA nanoparticles. So with the dextran cap and chitosan cap and the emulsified in C and D, you will see more particles appearance in the SEM image. And also there is a variation in the antioxidant activity of curcumin loaded PLGMPs with a different capping agent. We already saw it in one of our earlier slides. So this histogram gives an idea for the antioxidant activity of raw curcumin, TPGS, and the PLGA nanoparticles, curcumin loaded PLGA nanoparticle with the capping agent with the emulsifier. You will see a variation between 46 to 84 capping agent and also with the emulsifier. And the confocal laser scanning fluorescent image, it tells you the intracellular accumulation of curcumin loaded PLGMPs in the breast cancer MCF cell lines. So you will see three different colors. The green one are live cells, the orange or yellow orange are the one which is affected by the curcumin, and the red ones are necrotic cells which finally dies away. So you will see more and more of red colored, that is completely dead cells when you have the tocopherol emulsified dextran capped curcumin loaded PLGMP. This has the highest ability. The same thing is observed with the fluorescent image with the Hext 33342 dye also. The earlier one is a double staining method where we have used acridine orange and it's a dual staining methodology. And the cytotoxicity profile indicates that the last three, the tocopherol with the curcumin loaded, dextran cap, tocopherol with the emulsified with the dextran loaded, and tocopherol emulsified with the curcumin loaded. They have the higher cytotoxic effect. And not only nanoparticle, we also ventured into nanofibers. So how do you, we already elaborated the way in which we prepared the curcumin loaded PLGA nanoparticle with three different capping agents, namely chitosan, dextran, and polyethylene glycol, and also with the capping agent TPGS. So this is with respect to nanoparticle. Not only nanoparticle, we also made nanofibers. This is obtained by electrospinning. Of course, we have PLGA with a different LAGA ratio, and you have the curcumin, and you have the electrospinning. By electrospinning, you use the medium of methanol and chloroform. So by electrospinning, you end up with the nanofibers of curcumin loaded PLGA. By controlling the parameters like concentration of curcumin as well as the PLGA and the applied voltage, and also the distance of source electrode from the substance, the temperature, the solvent. Of course, we have used the mixture of solvents. You can vary the composition. So when you are altering this, the nature of the nanofiber also changes. This was published almost 8 years ago in Colloids and Surface B. And these are the same images of the curcumin loaded PLGA nanofibers. The left extreme, the top one is the 78:22 ratio, LNGA. This is 69 and 31. and this is 61 and 31, where this curcumin is loaded in the PLGA nanofibers. And also we have followed the drug release kinetics. In fact, people who are working in the pharmacology department, they will look at both pharmacokinetics and pharmacodynamics. So we have done both, and we have looked at the drug release kinetics of the curcumin. The curcumin is a drug here, and when you follow the kinetics and fit into various models, whether it's a zero order, first order, or pseudo first order, or the second order, or Higuchi, or Horseman, different mechanisms have been proposed. When you plot it and find out the R-square value, and the one which gives close to the one gives an idea which closely fits into the mechanism of the drug release. So we have fitted into different models and found out the Higuchi model fits into the drug release kinetics of curcumin from the PLGA nanoparticles. You could see from the nanofibers the value of 0.94, 0.97, 0.99, whereas in the Course-Meyer peppers it is 0.86, 0.93, and 0.98, which clearly demonstrate that Higuchi model is the appropriate mechanism which we can attribute to the way in which the curcumin is released from the PLGA nanofiber. And subsequent slide shows the plot, the Rooty versus the concentration, that's a Q. So you could see the R-square value of 0.94 and 0.86. So the earlier one is the course Higuchi, the second one is the Course-Meyer peppers. I'm not going into the details of how these various mechanisms are proposed. That itself will take one full session. So I'm sorry, I'm not going to the details, but it's enough to understand that there are various mechanisms where we can fit into the drug release and see to which kinetic model this fits in. So here we have found out that the Higuchi model is the appropriate one in a different ratio. 61:39 also we are able to get R-square value of 0.99, and whereas in the Course-Meyer it is 0.98 only, which again indicates that Higuchi model is the more appropriate mechanism by means of which we can explain the kinetic release of the curcumin, that's the drug release from the PLGA nanofiber. So to conclude, so we have what we achieved in this nanomedicine, that is the drug obtained from the naturally occurring turmeric and use it encapsulated in a nanoparticle as well as in a nanofiber, and looked at its efficacy as an anti-cancer agent, particularly the breast cancer agent. In fact, we have done it for other cancer cell lines also, but we could get the best result with the HCF7 breast cancer cell lines. And there are reports, in fact, recently we have written a book chapter where we have explained the use of curcumin in 12 different type of cancers, where people have reported the efficacy of curcumin in containing the growth of different type of cancers, like from leukemia to the brain tumor. So to conclude, what we have done reported so far from our own researches, we have could demonstrate the synthesis of TPGS emulsified curcumin-loaded PLGA nanoparticles with the different capping agents like curcumin, dextran, and polyethylene glycol by solvent, water, oil, and water emulsion technique. This is the first time these three different combinations are tested against the breast cancer MCF cell line. One is the PLGA TPGS dextran curcumin, PLGA TPS chitosan curcumin, PLGA TPGS polyethylene glycol curcumin, and the The in vitro curcumin studies occurs in a sustained manner up to a period of 30 days, as we indicated in our earlier table as well as in the graph. And the curcumin loaded nanoparticle exhibited increased preferential cytotoxicity to MCF7 when compared to free drug. You can ask the question, when curcumin itself is anti-cancer, why do you want to load it in a nanoparticle? We have demonstrated the higher efficacy of the curcumin when it is there in a nanoparticle rather than as a free curcumin. And of all the nanoparticle, we have found out dextran capped drug load curcumin loaded PLGMPs has the highest activity when compared to chitosan and peg cap. When it is emulsified with the TPGS, we get the highest activity. The relative efficacy of the drug carrier system varies in this order, with the tocopherol dextran, tocopherol with the chitosan, tocopherol with the chitosan, and tocopherol with the peg, and dextran with the chitosan, dextran with the curcumin, chitosan with the curcumin, and polyethylene glycol with the curcumin. So this publications over a decade, and we could see so right from 2011, Jano Biomedical Nanotechnology, then Advanced Science Data, Advanced Material Research. Then you have the collagen surface, B and biointerfaces, then current drug delivery. Then your Journal of Material Chemistry B, the RSC publication, then collagen surface B. And not only that, we also demonstrated the insulin as a oral delivery using this PLGA that we have published in International Journal of Medicine in 2015. And not only with the PLGA, also with the PLGA for the multi-drug delivery. And again, International Journal of Biomicromolecules 17, and carbohydrate polymers in 2017, International Journal of Nanomedicine in 2019 for amphiphilic copolymer nanoparticle for TB drug delivery. And then for tuberculosis drug delivery in 2019, Journal of Industrial Engineering Chemistry, and finally in International Journal of Biomicromolecules 2020, where the most of the work what I have presented now from the work what we have reported in this paper in 2020, the remarkable role of emulsifier and chitosan, dextran and peg as a capping agent in the enhanced delivery of curcumin by nanoparticle in breast cancer cells. And so also we have a few patents. In fact, our group has a maximum number of patents in the University of Madras, out of which 3 of them are in this nanomedicine. So the most all this work in the nanomedicine were done by 2 of my graduate students, one Dr. Malathi and Dr. M. Gajendran. And also we have tried out, it is an add-on thing, where we are using nanomedicine for chronic wound healing, where the doctors have advised, particularly for diabetic ulcer. So we have made use of the Siddha drug. We have combined, carried out research with the Siddha doctor, who is basically my colleague who did his PhD from zoology department, and he is practicing Siddha as a full-time Siddha practitioner. So he has a Siddha ointment, which is basically from copper base. So where the wound like this, particularly ulcer in the foot for a diabetic patient, even with a powerful antibiotic, it doesn't cure. So it takes months together, sometimes years together, and finally it gets infected and gangrene sets in, and the doctor advises to go for amputation of either the finger or the limb itself. So in this, we could apply the Siddha drug, that is Siddha Kalimbu, over a period of 3 months, and it got it cured for he has done it for several patients, but we have taken 2 patients for this patent, and whose age is 62 and 65 years with the known diabetic thing. So in both the cases, the wound got completely cured in a period of 3 months. Okay, so I would like to acknowledge the work done by Dr. S. Malati, who has done all this anti-cancer agent, and Dr. Rajendran and Gajendran, who did some cancer work and most of the TB. Malati has done with both TB and cancer, and as well as with the anti-diabetic with the insulin as oral delivery. And we would like to thank the University of Madras for all the infrastructure facility and the Department of Biotechnology, New Delhi, for the financial assistance. And also I would like to thank all of you, the participants, the faculty, the HOD of the Vishwa Vishwavidyalaya, the participants who are attending this faculty development program, the Vice Chancellor, the Dean, the Director, the professors, and all the faculty, and the coordinator, Mr. Venkatesh Jayakumar, for inviting me to and sharing our research on behalf of the Royal Society of Chemistry, South India. Once again, I would like to thank you all for your patient listening. If you have any questions, you are welcome to ask. I'll be happy to answer all your queries. Thank you.
Thank you, sir. Thank you so much for this valuable session. Now the session is open for the question answer. Anyone is having any questions, so they can raise the hand or open, or you can write your question in the chat box also.
So,
Sir, I would like to ask a question.
sure.
Subramanian sir, thank you
Yeah.
for very effective delivery, very relevant, very important, and very nicely you have presented the things. No question means people understood it, but I have one query.
Dr. Kavita, I would like to interrupt. There are 2 ways. If there are no questions, either people didn't understand or people have understood everything.
No, sir, it was very effective delivery.
Okay,
I'm sure people have understood it, but I want to ask, sir, how we can increase its solubility in water as it is soluble in organic solvents only?
Yeah.
Is there any way to increase its water solubility?
Yeah, so that's a good question. The thing is, it's a general phenomenon. Can you dissolve gold or silver in water? Suppose if you take a gold metal or silver metal and grind it into small powder and put it in water, will you get a solution? No. Everything will settle down. The same thing, you make it into a nanoparticle. You take a gold nanoparticle or silver nanoparticle, you will be able to get almost a true homogeneous solution. That's the beauty of the nanoparticle. The size, the entire chemistry and the property varies with the dimension, that is the size and shape. And those physical and the chemical properties changes. The so-called insoluble copper or silver or gold metal, once you make it a nanoparticle, it completely becomes a true solution. You will be able to get the spectrum in solution. That means you are getting a UV visible spectrum in a solution mode for a nanoparticle in a aqueous or a non-aqueous medium, right? So the one way of increasing the solubility of a material is to make it smaller and smaller from a millimeter size to micrometer to nanometer. You are increasing its solubility by several fold. So in fact, in the market, I'm not sure how many of you are aware of this fact. India is the largest producer of curcumin in the globe. So turmeric, of course. So we don't make nano curcumin in our country. There are companies in, of course, in Kerala, and of course in part of Tamil Nadu and Gujarat and Delhi. So they do solvent extraction. It's nothing but your oxalate extraction of curcumin from turmeric. You get the curcumin as a bulk form, pack it and send it to United States or in Europe, where they converted it as a nanoparticle and put it in a capsule and sell it 100 capsule, each one carrying 100 milligram of curcumin for a price of $60 to $80. So that is the value addition, because the raw curcumin, when it is made into nanoparticle, the solubility increases. The same nanoparticle is embedded in a polymer nanoparticle like PLGA. You are still increasing the water solubility, and thereby you are increasing the bioavailability. The smaller the size, the higher is the solubility. This is a simple formula. Is it clear?
Yes, sir. Thank you, sir. Sir, you have done lots of pharmacological studies on different chronic disease patients.
Yeah.
So in which chronic disease you found it most suitable?
No, as far
For
as
treatment?
no, actually, this control list we have taken up for 3 different type of diseases.
Yes.
One is actually when we submit to the project to DBT, it is for tuberculosis. As I said, this is a neglected disease by Western countries, because hardly you see any patient in developed countries with the TB. But it is more prevalent in a tropical country like India, China, or in many of the Asian countries. So not much work has been done in Western countries as far as TB is concerned. That is why we took up, there is something called, I am not sure how many of you are aware of this terminology, chronic disease and the what you call as a chronic. If it is a chronic, that means it is not cured over a very short span of time. You have to treat the patient over a longer period of time. Yeah, to get the to cure the disease. Okay, so TB, cancer, all these come under the category of chronic disease. That means the patient has to take the medicine over a period of time, weeks, months, sometimes years, sometimes throughout your lifetime. Okay, so that is where the patient compliance comes into picture. That means if you are asked to take 3 capsules or 5 capsules every day 3 times, you get really irritated. First few weeks, okay, you won't mind taking. For months together and years together, if you are asked to take, you get really irritated. You don't want to take the medicine. Then after some time, you discontinue it and say, I am all right. Why should I take the medicine? There, the patient compliance is not there. To achieve patient compliance, you should be able to give the medicine with a lesser dosage, with the shorter span of time. That is what we achieve in a controlled drug delivery. So TB is one of the chronic disease where the treatment normally it takes 6 months to 1 year, in some cases for years together. So if you can reduce the span, the treatment period, you can really get good, get good result. So the one way is embedding both hydrophilic and hydrophobic drug molecule of TB, that rifamycin, INH, and pyrazinamide in the same polymer matrix and give it to the patient, which will be released over a period of time, so that the dosage will be minimized and the side effect also will be minimized, and you can also achieve the patient compliance. This is the first set of drug or anti-TB. Then anti-cancer, we have used curcumin. For anti-diabetic, we have used the insulin incorporated polymer matrix. Of course, we have done with the not with the human trial, but with the animal model. So we have induced the diabetes in a rat, and we could, of course, give the insulin and see that it is work. People who are aware of the insulin, you have the short acting and long acting insulin. The short acting insulin was able to give a very, that is zinc insulin, could give a very good result in the animal model. Even for wound healing, when we induce the diabetes in the rat, when you make a wound on its back, within a week's time, when you apply the Siddhar Galimbo, so the wound is completely healed. Okay, so this is what, and of course, for the leprosy also, we could do it in animal model, so we didn't do it the human model. So all this we could get a very good result. Only thing is the period is very short, and of course only few years. So if we could continue the work, we will be able to demonstrate more results and more optimized nanoparticle formulation for treating these patients, which you can definitely go for clinical trial. This is our idea. Of course, as far as wound healing is concerned, already it has been successfully clinically trialed, and several patients within the country and outside the neighboring countries like Sri Lanka, Malaysia, Singapore, and all these patients, they came to Chennai and they got the treatment and got cured from this diabetic ulcer. The case histories are available.
Thank you, sir.
Respected Professor, it's a remarkable work, Professor. It's a mind-blowing work. Actually, I have one doubt, Professor.
Okay, thank you.
In the case of in the PLGA synthesis, so we are using tin chloride and tin octanoate as a catalyst.
Yes.
So if we, what about we can use tin iodide in the place of?
No, actually, what I would say is these are the first two, and also there is another question which you could have asked. Will that tin will remain in the polymer after the synthesis over? No, because yes, after repeated washing, we will remove all the catalyst. Catalyst itself is used small quantity. Even that catalyst will be washed away during this process. So no tin will be there in the PLGA in the final, because you are giving to the living system. So tin should not be there. So that is totally taken care of, number one. Number two, both the first report was with stannous octoate. That gave a very good result. Subsequently, stannous chloride was used, and we followed it, and we were able to get good result. After that, as I said, you could see only the patterns. So there is no complete detail. One can try. I'm not saying you should not try, because just because we have not tried doesn't mean it will not work. So it may work, it may not work. It has to be tried out. There's absolutely no harm in trying out with the other stannous compounds. Yes. Of course, we should optimize the condition and see whether we are able to get the same result or with the lesser efficiency or higher efficiency. We have to do the experiment and find out.
Yes, thank you, Professor. And another doubt, Professor, in the synthesis of dextran capped curcumin, DC-PLGA nanoparticles, and you are using a sonicator probe for sonication purpose.
Yes.
What will happen if we keep the solution, the reaction mixture, directly into the sonicator bath? instead of
Ah.
sonicator probe?
Yeah, you are. That's a good question. See, usually the most cases, both the drug and the carrier are sensitive to temperature.
Yes.
That is why PLGA we are keeping at minus 40 for storage. Or even at a fridge, of course, sub-zero also you can keep it for a longer period. But once you take it out, you incorporate it. You incorporate the drug molecule and use it. But what happened? Of course, we with the couple of slides I have not shown. We have done the TG and DSC of this PLGA. See, the point here is you cannot keep the PLGA for a longer time at room temperature. That is why you keep it at lower temperature and you incorporate it and then you distribute it. So this is what normally people do it. Of course, usually the thermal heating is not advisable. Number one, it will lead to the degradation of the polymer matrix. Number two, a simple logic, any in a simple gravimetry experiment, after precipitating, what you do is you will heat the solution so that the particle aggregate and you will be able to filter the precipitate without any problem. That means you are aggregating the material. The same thing occurs if you are heating a nanoparticle, then the aggregation occurs and you will end up with a micro-sized particle rather than a nano-sized particle. So all these problems will occur if you are going to give a heat treatment. All these can be eliminated if you are using a sonication, not with the bath, but with the probe. Because bath also, the temperature will go. Of course, you can control the temperature, but bath you don't get this kind of result. That is why we optimize the thing with the probe sonicator rather than a bath sonicator, which is available in almost all the labs. Even in many of the undergrad labs, they will have the bath sonicator, which cannot be used for this purpose.
Thank you, Professor. Very kind of you, Professor. Very kind of you.
Any other question from the participants?
One more question is there.
Yeah, sure.
Hello, Divya, can you hear me?
Yeah.
Dr. Subramaniam, I have a question.
Sure.
Does curcumin show selective absorption of the cancer cells?
Yeah, because that we have demonstrated with the cytotoxic experiments, both with the normal cells as well as the cancer cells. So where the RO, because of the reactive oxygen species present there in the cancer cell, the absorption is very high in cancer cell when compared to the normal cell.
Okay.
That is why you get a selective absorption and also the EPR effect. So these two things dictate the selective accumulation of curcumin in the cancer cells rather than the normal cells.
Okay. Okay. Thank you, sir.
Thank you. Any other question? Attendees? No question. Thank you so much.
I think there are two questions in the QA. Oh.
So the same question actually.
No, Mr. Shadab Sheikh. I think is typing an answer. That's what it says. Shadab Sheikh.
So that is
Oh, that was the question.
yeah. It was a question if you compare it.
Yeah, if you compare it now, that's fine. That's fine. Okay, good. So still, I have given my mail ID in my first slide. So probably people who have more questions, I'll come back to my first slide. bala2010@yahoo.com. If you have any questions, you are welcome to communicate to me through email. So you are most welcome, which I will be glad to answer your questions.
Thank you so much, sir. Thank you for this valuable talk. Now I request Dr. Venkatesh sir for words of thanks. Kavita ma'am for the words of thanks.
We are very thankful to Dr. Balasubramanian sir for very effective, very nice delivery, and to all participants who have attended it. And I request all the participants to be with us every day. We will share the schedule in the WhatsApp group. Thank you very much. Thank you all.
All of you are requested to fill the feedback form, and the feedback form for all the days are compulsory to get the certificate for the faculty development program. All the attendees are requested to fill the feedback form on a daily basis. After every session, the feedback link will be in the chat box. Thank you.
So thank you very much.
Thank you so much, sir.
Thank you. Thank you
sir.
all. Thank
Thank
you
you,
all.
sir.
Respected Professor, it's a very inspiring talk and very great
Thank
work,
you.
Professor.
Sure. Bye.
Thank you, sir. Shada sir, please close the meeting.
Yeah, I'm sorry.
Thank you. Thank you.
All the attendees are requested to join the WhatsApp group also. You'll get the information of the WhatsApp group of the FD. Whoever have